Good morning, everybody. All right, so take out your guided notes. We're going to start Unit 0, and this is our foundation for our scientific thinking in AP Bio. And it's not just content you're going to memorize, but these are skills and tools you're going to use throughout the year, specifically in lab reports and also in any science course you take in college. All right, so remember you're going to be actively filling in the blanks throughout the video. And in this unit, we'll cover experimental design, which is this video. Eventually, we're going to get into graphing and then statistics through chi-square analysis. All right, so this is Science Practice 3.2. And so this is what's going to be on the AP exam. So it's 3.8 states that you have to identify experimental procedures aligned to research questions. And so notice here how you're going to be identifying independent variables, dependent variables, identifying appropriate controls, and then justifying why you use these controls. All right. The AP exam is not going to ask you to define experimental design. Instead, it's going to ask you to analyze experiments or experiment scenarios and then explain why they work or don't work. And so in the AP test, you are going to do experimental design, like I said. So this means identifying the variables, explaining the purpose. You're also going to be asked to graph things in the AP exam, and then also make conclusions and analyze the data. All right, so what is experimental design? So let's start with our basic definition. So experimental design is the process scientists use to plan effective experiments that utilize the scientific method. And we'll get into the scientific method, but think of experimental design like planning a road trip, right? You won't just start driving randomly hoping to reach your destination. You're going to want to plan something, all right? And so notice how there's a flowchart for the scientific method, because all good science starts with an observation and a testable question, right? So testable means that it can be answered through experimentation. And so look at these three examples right here. And so this is from Newton, the first example. The apple falling led to gravity theory. Fleming's moldy petri dish led to penicillin discovery, which was an accident. And then Einstein wondered about the speed of light, led to relativity. All right, so each started with a simple observation, and that became a testable question. All right, so what's an observation you guys have recently made? Try to think about that, and then we'll continue. All right, what do you need for your experimental designs, right? We said you need one, so what do you need? All right, so in experimental design, notice how first you need a hypothesis, and this is a testable prediction. So this is your guess on what's going to occur. It's your educated guess. And then second, you're going to need your variables, the independent, dependent, and your constant. And then third, you need controls, right? So this is your baseline. A hypothesis is a prediction or a possible answer to the problem or a question you are attempting to answer. Again, your educated guess. And so it's always going to predict the relationship between the independent and the dependent variable. It's always a cause and effect relationship. And so look at the example right here. And notice how the hypothesis is always going to explain what's going to happen. It's going to be clear and understandable. It's going to be testable. It's going to be measurable. And again, it's going to have your independent and dependent variable. All right. So this example is discussing the color of light in an environment during a studying will decrease a student's performance on a test. So this is your answer, right? What you think is going to happen. And so notice how it's specific, it's testable, and it makes a prediction on the relationship, okay? So how can you make a hypothesis? How can you formulate a hypothesis? So first you have to identify your independent variable from your observation. Second, you have to think about your dependent variable. And then third is what is the relationship you expect to see, right? Because there's always going to be a relationship. Before we move on, just practice. So if we want to study whether caffeine affects test performance, what would be our independent variable? All right, so pause it right now. All right, so if you think that caffeine will change your performance, then caffeine would be the independent variable. It's what you're changing. So whether or not you drink caffeine, whether you drink caffeine, or the amount of caffeine you're drinking. And so what would be your dependent variable? What's something you're going to be measuring to see if the caffeine is working? And that should be the test scores, right? All right, so let's move on to types of hypothesis, because you can have two types of hypothesis. There's null hypothesis and alternative hypothesis. Null is represented with H naught, which is like H and a little like 0 at the bottom. And then alternative hypothesis is HA. All right, and so null hypothesis states that there's going to be no observed effect in the experiment. And so you're predicting that the independent variable has no effect on the dependent variable, and you're assuming that your treatment's not going to work, right? There's not going to be any statistical difference between your data. And so this is the conservative approach. You're skeptical, until you're being proven wrong. And so the alternative statement is the opposite, right? So you do predict that there is going to be an effect, a statistical difference, right? And so an alternative hypothesis or all hypothesis should be written in if, then, because statements in AP Biology. And all hypotheses are simple because you're basically going to say that there's going to be no effect. So, for example, if I study for my classes, then I will achieve higher grades, versus studying for my classes will have no effect on my grades, right? That's a null hypothesis. All right. And so here's basically a little summary of that. You want to use the if then for the example. All right. Another example of a null hypothesis is if you're testing two types of water and seeing if the type of water is going to affect plant growth, then you would say that there's going to be no statistical difference or relationship between plant growth and the type of water you're using. All right, so go ahead and now practice these together, and when you're finished, check with a partner. All right, and so to test your hypothesis, you also need something called a controlled experiment, and we'll talk about that in a second. But controlled experiments are the things that you keep constant. All right, and so for example here, the independent variable is going to be something that's being manipulated or changed. All right, and so here you see that the thing being changed is the plant liquid. So they're either using soda, juice, or water. And notice how they're only changing the type of liquid. So you want to limit the thing you're changing to one thing per experiment. And so the dependent variable is going to be the response, right? So what's going to be measured throughout the experiment. And so here it's the plant height. And so here we're wondering, does the plant height change based on the type of liquid? And so your control here is anything that stays constant. So there's going to be basically the same type of liquid being used, the same type of plant, the same pot size, in order to ensure that nothing else is affecting this experiment or nothing else is causing there to be an effect. All right. And so here's a summary of that. Again, the independent variable is what you're going to manipulate or change. And so if we said that the independent variable is what's going to change, then what do you think is the independent variable in this scenario? So a scientist is doing an experiment to determine how light intensity affects plant growth. They set up 5 plants in different intensities of light and measured the growth of each plant. All right, so pause the video and predict what the independent variable is. Again, the independent variable is what's being manipulated or changed, and so here we see the light intensity. The dependent variable here would be the measure of growth, so the growth. Because a dependent variable is what's going to change, all right? It's going to be what responds to the independent variable. So again, you want to try to remember independent causes, dependent responds, and then the control stays constant. Okay? So go ahead and practice identifying independent and dependent variables in these scenarios. All right, and we kind of already talked about constants, but constants are what's going to remain constant, right? So it's going to remain the same throughout the experiment, and this is again to ensure that the changes are due to the changes in the dependent variable are being caused by the independent variable and not anything else. All right. In the experiment, does sunlight affect plant growth? What would be some controlled variables you'd like to have here to ensure that the sunlight is the thing that's going to cause a change in the growth of the plant? So think about that. All right, so let's move on to types of groups found in experiments. All right, so we're moving on to types of groups in experiments. So what are experimental groups? Test groups receive various amounts of or types of independent variable treatments that are different than control groups. And positive control groups receive the normal amount of the independent variable. And negative control groups do not receive the independent variable. And so you want to go ahead and fill in these definitions. And we want to remember that all experiments will have a control, but not all experiments will have a positive and negative control. So what is a positive and negative control? And so we're going to compare and contrast them. A negative control, like we said, is not going to be exposed to the treatment. We don't expect the treatment to have an effect, right? Nothing is going to happen because there is no treatment. A positive control is going to be exposed to the treatment that we know is going to produce an effect. So we want to make sure we have the positive in order to make sure that there is going to be an effect when that's going to take place. And so the way you could think about this is, like, for example, if you're taking medicine for your headache, your negative control, like for example, I can give someone a sugar pill that's not going to get rid of the headache. And then the positive control is going to be like the actual medicine. It's going to relieve that headache, right? And so if we know if the positive control doesn't work, then something is wrong. And again, if the negative control works, then, it's a placebo. Sometimes people could think it works, but again, nothing should occur. All right, and so here's another example. The rate of a plant growth is directly related to the intensity of light received by the chlorophyll molecules in the thylakoid membranes of the chloroplast. So plants normally receive 8 to 10 hours of sunlight depending on the season. So scientists expose different plants to different light intensities and measure plant growth. So the positive control here would be receiving the sunlight, right? We know it's going to make the plants grow more. Negative would mean receiving no sunlight at all, since we know that not having sunlight affects plant growth, right? And then the experimental groups, you could change the amount of hours these plants are receiving the sunlight. All right, and so you go ahead and try. We talked about the pill already. So you go ahead and try the solar panel practice problem, and then there's also a pesticide practice problem, and then come up with positive and negative controls for both of these. All right, and the last thing you're going to do is you're going to come up with a null hypothesis and an alternative hypothesis for each of these questions. You don't have to do a graph because we haven't gotten into graphing yet. But go ahead and do your null hypothesis, your alternative, and any other things you'd want to have a controlled experiment. And then we could go over the answers.